Our research question is, what is the digestibility of different types of proteins, beef, salmon, and Beyond Meat, as tested by their change in mass after a 30-minute submersion period in simulated gastric acid solution, and the remaining mass's effect on the temperature of water tested by a calorimeter? Our independent variables were different types of proteins, either beef, salmon, or Beyond Meat, and we had two dependent variables, which the first was the change in mass in ounces after simulated gastric acid submersion of the different types of proteins using a scale, and the second was the change in temperature in degrees Celsius of the water in the calorimeter after 5 minutes using a thermometer. Our theory is that during digestion, the stomach breaks down food using gastric acid in a process called chemical digestion. The gastric acid is composed of hydrochloric acid, which is what breaks down the food. Our lab focuses on digestibility as a change in the food's mass as it's broken down.
To prepare for the experiment, the protein should all be cooked in the oven at 190.5 degrees Celsius without any seasoning. To start the experiment, measure out 2 ounces of protein and place it in a bowl. Then measure 150 milliliters of simulated gastric acid and pour it into the bowl. Once the acid is poured in, start a 30-minute timer. Once there are 12 minutes left on the timer, stir the meat in an attempt to replicate the churning process. Once the timer runs out, drain the gastric acid to separate the undigested meat. Then transfer the meat onto the hot plate, which is at 180 degrees Celsius. The meat should be on the hot plate for 9 minutes to evaporate as much of the remaining acid as possible. After 9 minutes, transfer the meat into the calorimeter as quickly as possible. The calorimeter should have 150 milliliters of water with the thermometer measuring its temperature. Check the temperature of the water at the very beginning and then after 5 minutes. Repeat this process 5 times for each meat to gain enough trials.
Some of the variables that were controlled well in the experiment included the amount of time that each of the meats was simulated to be digested for, the amount of time on the hot plates, and the amount of time in the calorimeters. As well as this, the initial mass of meat, type of acid used, and the amount of water in the calorimeter were all controlled well. On the other hand, variables that not controlled too well consisted of the actual temperature of the proteins. Even though they were heated for the same amount of time, we didn't ensure identical temperatures.
Our data shows that beef was digested best, as it had the highest mass decrease, although it decreased by only 1/100th more than the Beyond Meat. Realistically, the Beyond Meat should be the easiest to digest due to its higher fiber content since it is created out of vegetarian foods. This allows for Beyond Meat to be digested in under a day, unlike the beef and salmon, which have lower fiber contents. In addition, proteins and fats contained in meats are made of complex molecules which take longer to break down in the digestive system. On the other hand, Beyond Meat contains simple molecules which are easier to break down. This shows how our data might be skewed. This will further be discussed later in the video by the ANOVA test we ran.
When looking at our temperature graphs, the beef had the smallest average change in temperature because of its mass decrease when digested, so its final mass was the smallest or most digested. Our data displays beef as the most digestible protein type, although it does not reflect real-life circumstances as explained previously by Rocio. In terms of limitations, the different distributions of fat and protein within the same variable could affect digestibility. The size of the food chunks is not realistic as we would normally chew our food before digesting it. The lab is unrealistic in terms of a real-world digestion process. The amount of gastric acid is not realistic to the quantity found within our stomachs, and the time that the meat spent in the acid is also unrealistic, as a human usually spends 6 to 7 hours digesting food. We had to make our own funnel system with a styrofoam cup and a hole in the bottom because of a lack of supplies. Our group worked well together, and we divided the work in a way that was efficient. The diversity of proteins provided variety, reflecting different diets such as vegetarianism, pescatarianism, and the standard diet. When simulating the stomach, we took into account the churning motion of the stomach when digesting food. We were consistent with our hot plate usage, meaning the temperature and time any given meat was on the plate was the same. Our procedure was flawed as our initial idea of calorimetry and what a calorimeter measured was incorrect. The calorie type we were looking for was not measurable by the device we chose to use.
When running the statistical ANOVA test for temperature, it shows that our p-value for temperature is 0.59, which is much larger than 0.05. This implies that there is not a statistically significant difference between the 3 independent variables. Although the error bars between the salmon and the beef do not overlap, meaning that there is a slight statistical significance between the two. The ANOVA test for the mass values gave us a p-value of 0.41 once again, meaning this data was not statistically significant. Essentially, there is no real difference between the protein types and their digestibility, and therefore no one diet is better for the body than other in terms of digestibility.
Now for improvements to procedure. If we were to repeat this experiment, we would use a different method of calorimetry and have a more effective draining process when isolating the undigested food from the acid after 30 minutes. We would add a strainer to our materials list to make the straining process faster and less messy since we needed to create our own funnel. While working in an efficient manner for the time given for this experiment, to make the experiment stimulate the digestive process of the stomach more realistically, it would be better to let the food sit in the gastric acid for a time period which would represent the human digestion, something close to 5 to 7 hours. Also, we would use larger amounts of meat in each sample to more accurately represent a human food intake and place the food in a processor beforehand to simulate the chewed up food more realistically. Furthermore, for a more realistic churning motion, rather than simply stirring with a glass stirring rod while the food was in acid, we could have it squished inside of a bag that would sustain the effects of acid, or use a contracting bag.